Welcome back, welcome back, welcome back. All right, 6.2 is going to hit on enzymes, and enzymes are going to be a focal point that you'll see many times throughout the year. So, enzymes, they are a catalyst. They're going to speed up a chemical reaction. And um, a catalyst itself is not used up, it's just going to be used. So, you may think of something that you use every day, maybe a lock and a key. Uh, when you're finished opening your lock, you don't have to replace the lock or key. It's used but not used up, and it's going to speed up the reaction of which things occur. And you may think, well, that analogy really doesn't work well. You have to use your key to get in your house. If you waited 500 years, you'd eventually be able to go right in your house, right through that doorway, without any additional effort, because things will break down and decay over time. It would take a lot of time for that to work. But because of a catalyst, you can do things much faster. So enzymes are biological catalysts. These are proteins. Once again, all enzymes are proteins, but not all proteins are enzymes. So this enzyme is going to speed up a chemical reaction, and it's going to do that by really lowering the energy needed to get from reactants to products. Here we have a reaction where we can see sucrose being broken down using the enzyme sucrase into glucose and fructose. So this is an example of an enzymatic reaction. And once again, enzymes end in ase, and oftentimes they're named for what they're breaking down. This is sucrose. You can guess what amylase breaks down. That's amylose, which is starch. You can guess what cellulase breaks down. That's the cellulose sugar. Yes, that large polysaccharide that we've already talked about. So you get an idea of enzymes that are going to really lower the energy needed. So the energy needed is going to be called the energy of activation or activation energy. Both of those are referring to the same thing. This diagram is one that you should commit to memory. I recommend you stopping and drawing it. And this is showing AB and CD that are going to be going through a chemical reaction, and it's going to end with AC and BD. Notice that we have higher energy, free energy to start with, lower free energy to end with. The energy of activation, so the activation energy, from getting to here, we need this much energy to do that. So sometimes this mountain is enough that keeps things from going to disorder. An example is your textbook. The pages on your textbook, which are cellulose, they are flammable. They can go into lots of smaller pieces. But what keeps them from breaking down is that the activation energy is much higher than just what would be a normal everyday situation. And so your textbook is not bursting into flames. And so going from high order, going from this negative delta G to positive delta G, or at least towards zero, this process is going to be an exergonic process. This is going to be spontaneous. This is going to have higher potential energy to lower potential energy as we go through this chemical reaction. So this oftentimes is seen as a catabolic type reaction as we go through that. So let's go into this a little bit further. So to catalyze a reaction, you're going to speed up the rate at which something is going to occur. This is going to be a catalysis. So the way an enzyme works is it catalyzes a chemical reaction which lowers the activation energy. It does not lower the free energy. It lowers the activation energy. So what that looks like on a graph is this. So going from reactants to products. Notice that in the presence of an enzyme, the energy needed to get from reactants to products is much lower. So it will still occur without an enzyme, but it is easier for the enzyme to occur. This is the free energy. Notice that where the enzyme is, that it's the same amount of free energy with or without an enzyme. You have the same amount of free energy. However, the activation energy is lower in the presence of an enzyme, meaning that you can go from reactants to products at a much quicker rate with much less energy input. Think of climbing a mountain or a speed bump. Speed bump is way easier. It takes a little bit of energy to do that, but not anything like this massive hill or mountain. And so you use less energy. So explaining this overall graph, we have reactants, we have products, we have non-enzymatic. This is without an enzyme. This is with an enzyme. This is the amount of energy needed from the peak of that to here. That's the amount of energy needed to make this reaction go. Think of it as a barrier. Here is the amount of energy needed in the presence of an enzyme. So the difference is the advantage of having an enzyme there. And so in the end, we end up with closer to delta G is towards zero. We have definitely decreased the free energy. And we can see that by this. So the free energy on this measurement, here's the free energy at this point. So we've decreased free energy. But with or without an enzyme, you're going to decrease the free energy. The time is longer whenever you have no enzyme, much faster with an enzyme. So if you look at an enzyme itself, an enzyme is going to have some characteristic properties. And it's going to act on something called a substrate. So let's go ahead and talk about these structures and identify them in just a moment. So this structure, this is representing all the different molecules involved. And we know that since they're all enzymes are proteins are at least 100 amino acids long. So these are big. And here you have this little bitty substrate. This is the thing that's going to be worked on, that's going to have the reaction done. It's going to have a very, very, very specific location that it'll fit. Only fit there. And when it fits there, this area is called the active site. So this blue, it would be all the enzyme. A very specific location on the enzyme would be the active site. The active site is the most important part of an enzyme, because if it is altered, denatured, if it is flawed in any way, it's not going to work. And think of a lock and a key. They're very, very fitted for each other. If you change one of them, you're not going to get the product. And so this is going to be the enzyme here, and here's the substrate. The substrate is going to bind in the active site of the enzyme, and now this is going to be called the enzyme substrate complex. This is just talking about the intermediate that occurs with this. So let's make sure that we've covered that. So we have the substrate enzyme complex, the active site, which we've all identified. So if this is going to be the enzyme, this would be the substrate. This would be the substrate enzyme complex. It would break it apart, and you would eventually have the same enzyme, and you would have an altered substrate. So induced fit. This is adding to the specificity of this. So the way induced fit is, it's really pointing to, at the active site, you're going to have chemical groups that are going to be working with each other to make this a really precise fitting, opposed to just kind of like you're wearing your dad's suit. Instead, this is going to be like you were tailored just for you. It is perfectly fit, and it's rearranged just in position to maximize the likelihood of the chemical reaction occurring at the best rate. So this would be a good list for you to have. And this is with the enzymatic reaction. This is going to be the advantages of the active site, what it does. So it's going to orient the substrate, kind of think in your head of what the substrate is. And if you can't, then take a moment, look back, see what the substrate is. And I highly recommend drawing this. So it's going to orient the substrate, so it's going to put it in the right position. So now it's in the right spot. It's going to be essentially working on it in itself, causing it to be ready for its reaction. It's going to provide everything right, that microenvironment. And it's even going to covalently bond. This covalently bonding piece, this is pointing to that induced fit concept that's here, just to make sure that we reiterate that the covalently bonding of the substrate is going to be key and making sure it is poised in the right spot. Imagine trying to open a door with the wrong end of the key trying to be entered. It's quite silly, and you would probably stumble around till eventually you'd randomly find it. This enzyme is going to help orient, guide it in, and then it's going to work perfectly. So let's take a look at these substrates. We can see two different substrates, this red and green, that are going to fit perfectly in this enzyme. And notice how perfectly honed it is. So now that it has gone through and it has bonded, this is now called the enzyme substrate complex, where we're going to have the actual chemical reaction take place. As a result of that reaction, that catalyzed reaction, you're going to have products which are going to be generated. The products are then going to be released away from the enzyme, and eventually you'll end up with this, the enzyme with its active site free once again, able to accept new substrates because it is not used up, it is not broken down in any way through a chemical reaction. So the rate at which enzyme is going to catalyze a reaction can be increased by the substrate concentration. If you think of the way, here's an analogy for you. Let's say that you're in school and you're playing hide and go seek in the school. And I know this is silly, but let's just go with this. All the classrooms are shut, so you're just using the hallways. Being able to find individuals that are going to pair up with that perfectly, that's going to happen. But you can increase the rate of the chemical reaction by increasing the substrate. So you would be the enzyme. You would be the finder, the one that's seeking individuals. In this analogy, you would also have the substrate being the people who are hiding. So if you increased substrate, if you increased the people that are hiding, the likelihood that you would find somebody would be higher. It would happen more rapidly. So when an enzyme is going to be bonded with a substrate, you're going to have the highest rate you can have is when the enzyme is essentially saturated. So enzyme saturation is going to be the reaction rate can only be increased by adding more enzyme. And so that's like adding more seekers in the hide and seek scenario. And so the more finders that you have working around, the higher amount that they'll find the substrate and will increase the rate. And so we're looking at ways of increasing the enzymatic rate of catalysis. And so you would increase that by saturation of substrate and then increasing the amount of enzyme. That's what would get us to have higher a rate. Now, the enzyme can be affected by a couple things. In chapter 2, when we looked at proteins as a macromolecule, we identified that enzymes are an example of this, of a protein, and you can denature these with heat, temperature. There are other things like salinity, radioactivity, electricity, but really the two that are most commonly spoken of and that show up on test are temperature and pH. So we have certain enzymes that are really going to do best at certain environmental conditions. And so those perfect conditions, those ideal conditions are going to be considered the optimal temperature and optimal pH. So take a look at this. Here we have the optimal temperature, and this is our body temperature, 37 degrees Celsius. We see the optimal temperature for this is right there. And so our enzymes work best right in that range. Matter of fact, if you get much further out, these enzymes are going to denature and they're not going to work at all. So zero enzyme work here. Or if you get too cool, these enzymes aren't able to work as well. Take something like the bacteria that lives in the hot springs, and let's say um, Old Faithful, or an example like that, where we have this water that is near boiling, scalding to say the least. Um, and we find that their optimal pH is going to be roughly 75 degrees Celsius. And so they're going to be most efficient. They're going to be running at their optimal speed at these temperatures. So this is pointing out that not all enzymes have the same optimal temperature. For those that are dealing with humans, they're typically around 37C, which is 98.6 degrees, our body temperature. And you have other things that are just in a completely different range. Like at our body temperatures, those things won't live at all. Here's another example of optimal conditions. And this is the optimal conditions for pH. So our stomach has a pH of 2. We have different enzymes like pepsin, which is going to be used and breaking down different digestive components. And pepsin has that optimal pH of 2. And so you can get the idea that around 2, it's going to work really well. With a pH of 4, 5, 6, it doesn't work at all. And so whatever you're trying to break down doesn't get broken down if it's outside of that range. You get something like trypsin, which is going to be used in your small intestine. This is going to be, this has an optimal pH of roughly 8. And so those optimal pHs that's the perfect condition of which they work. They work with most of efficiency, with the highest frequency of getting to product, from reactant to product. And so there is that perfect kind of Goldilocks moment of where they work. And so notice that there are different pHs depending on the different enzymes that are there. Is an example of those hot springs where you can see that bacteria that's growing, the algae that's growing on the inside, and it's doing perfect at this temperature, that cyanobacteria. So it's crazy that they do so well, but they do really well at a certain temperature. And that's not the temperature that we would do well. Now, when you look at enzymes, enzymes are going to have some other pieces that are sometimes embedded in it, like cofactors. Cofactors are going to be helpers that are going to help make sure things get done. And they're going to do this at a very specific spot. And so take a cofactor like zinc as an example. This cofactor is going to be part of, oftentimes, the active site. So think of the active site, this perfect part of the active site of this big enzyme. And this part of the active site that's here is actually made of zinc. So the enzyme is protein, but this little cofactor, this helper, is going to be zinc. If you didn't have that, you don't have that active site. And if you don't have that active site, you can't go through that enzymatic reaction, which means the process that's needed to be catalyzed, that's not going to happen. And so it's going to just be very slow. And sometimes the effect of stunning growth or many other factors of life. And some diseases are really played into not having certain things as part of this. And so if you're lacking that cofactor, you can't live life normally because those enzyme, that coenzyme, that part that's going to make that, they're missing. So most vitamins are going to work as coenzymes, and they're going to be the thing that really helps the enzyme do its job. The last points I want to talk about are competitive inhibitors and non-competitive inhibitors. So if you think of the structure of an enzyme, a competitive inhibitor, if this is the enzyme, once again, the competitive inhibitor is going to get in the way and it's going to bind in the spot that is the active site. When something else is going to bind in that, it's already full. Since it's not the right thing that's being broken down, not the right substrate, it doesn't make a change in this. It doesn't alter it and break it down. Instead, it's going to just be bonded in that spot, blocking this enzyme from doing work. So that is a competitive inhibitor because they're competing for the actual part of the enzyme that's trying to do work, the active site where things are going to bond. A non-competitive inhibitor is going to bond to another area of an enzyme, and it's going to cause it to have a distorted de- deflated a completely different active site. And so what would have worked in here perfectly doesn't fit because this active site has been altered due to a different part of this being misshapen. So many things like toxins, poisons, pesticides, antibiotics, they're going to be working on things like this. So here we have an example of this is just the normal um, substrate enzyme complex. Here we have a competitive inhibitor. It's in the way. This substrate can't bond, but it's bonded in the active site here, and this is not going to have a product. Notice that when a non-competitive inhibitor is going to bond this, it causes a misshapen active site. As a result of the misshapen active site, it's going to cause it to not do its thing. So most enzymes are going to be proteins that are going to be encoded by genes. A mutation of this is going to cause a change of the amino acid composition, and that in return will cause a change in the overall structure of the enzyme. All right, guys, that's 6.4. I look forward to seeing you soon.